6 Mutualisms. Mutualism is the association of two species, which is beneficial to both. A plus-plus relationship, see also Bronstein et al. 2006. Such species may live in close association throughout much of their lives, in which case the relationship qualifies as symbiotic mutualism. Some would argue that symbiosis involves the physiological integration between partners, but this criterion is not employed in most of the ecological literature. Examples include the many cases of microbial symbionts of insects, as in termites and their protozoan and bacterial associates. But mutualisms need not be symbiotic, with each species living a life of its own, as with plants and pollinators while, nevertheless, providing services to the other species. Mutualists may be obligate, a necessary association required for the survival and reproduction of a species, as with termites and microbial associates. They may be facultative, beneficial, but not essential for survival and reproduction. An interesting case of a facultative mutualism is the link between stick insects and ants in Australia, in which eggs of phasmids are dropped from the tree canopy to the ground. The capitulae on the eggs are attractive to ants, which carry eggs into their nests and to greater protection against natural enemies, Hughes and Westaby 1992. In Costa Rica ants disperse eggs, but do not carry them deep into the nest, Windsor et al. 1996. We will explore the rich array of mutualistic interactions, recognizing the evolutionary opportunities generated by reciprocal beneficial associations, and the many forms of mutualism involving insects. Then we will enter into the subject of the inevitable costs and benefits of these associations, and cheating the system. Mutualistic relationships have resulted in major adaptive radiations, which we explore with several examples, and then move on to approaches to modeling and how beneficial relationships may become complex and central to community organization, as well as in the practice of agriculture. 6.1 Mutualism is a creative evolutionary force. There can be little doubt that mutualism is among the most creative forms of interaction in nature. As an evolutionary force, the coupling of two species into a beneficial bond becomes a saltational event, frequently allowing, and enabling, a leap into a new adaptive zone, and the exploitation of novel ecological niches. Consider the evolution of the eukaryotic cell, formed by the community of mutualistic prokaryotic species, and the abundance of life derived from these unions, e.g., Lee and Frederick 1987, Margulis 1993, Margulis and Fester 1991, Sapp 1994, Werner 1992, Cavalier-Smith 1987, 2006. Union of prokaryotic cells resulted in momentous quantum evolutionary episodes of cellular innovation, Cavalier-Smith 2006, p. 969. Purple bacteria, using respiration as an energy source, colonized a prokaryotic cell to form mitochondria in the eukaryotic cell. In a similar manner, cyanobacteria capable of photosynthesis colonized protist cells which enabled photosynthesis in the eukaryotic cell and the evolution of plants, figure 6.1. The creative opportunities provided by mutualism have only expanded since the evolution of the eukaryotic cells. 4 new kingdoms of organisms have emerged, protozoa and other protoctista, plants, animals and fungi, e.g., Margulis 1993, and members of these kingdoms have combined in mutualistic associations with far-reaching consequences, enabling the emergence of large plants, herbs, shrubs and trees, and large animals, figure 6.1, Price 1991c, 2002a. Large plants could evolve in association with mycorrhizae, pollinators and seed dispersal agents, and large animals depend on large plants for food, with many sequestering microbial digestive agents in rumens or cc. Even humans enjoy mutualisms with many bacteria in their intestines, Ba Diaresis Head et al. 2005, and as probably with insects, the microbiome may contain 50 to 100 times more genes than the human genome, Dillon and Dillon 2004. Species themselves should be considered more as ecosystems than as single individuals or species. 6.2 The variety of mutualistic interactions. Insects have benefited from mutualistic associations as much as any other group, and perhaps more. 4. Example, insects have radiated into incredibly diverse adaptive zones, frequently utilizing nutritionally inadequate resources such as blood, sucking lice, or plant sap, aphids, or highly refractive substrates, such as cellulose in wood termites, and keratin in wool, antlers, horns in tortoise carapaces, e.g., Darup et al. 2005. Clothes moths and trogid beetles are assumed to require mutualistic species, but this is not yet described in the literature, although ctenids are known to utilize keratin in which fungi occur, Robinson 2004. It is interesting to note that feeding on fungi is widespread in the Tinioidea and perhaps it is a ground plantaractor of the group, Robinson 1981, indicating a long-term association with fungi that can digest material in rotting logs and keratin of various sorts, Robinson 2004. In fact, we see in Table 6.1 that the range of mutualistic types involving insects is bewilderingly diverse. One estimate of the frequency of mutualism suggested that 45% of insect species in the British Isles are likely to participate, Price 1984b, 1997. However, including insect species that are impacted by mutualism involves many additional species, either directly, as in Table 6.1, or indirectly through the evolution of the angiosperms, or the influence of mycorrhizas on host plants of herbivores, or the radiation of the herbivorous mammals. Thus, major adaptive radiations, such as the flowering plants, angiosperms, or the herbivorous mammals, artiodactyls, parasodactyls, lagomorphs, rodents, etc., created new resources on which insects themselves could radiate. There are many ways to categorize and classify mutualistic associations, but we have chosen to emphasize ecological relationships, dividing them into 3 major kinds. There are exploitative mutualisms, or nutritional mutualisms, in which, by acquisition of a mutualist, an insect gains the ability to exploit a new resource, from which a novel adaptive radiation may develop. Here the nutritional value of the food source is incomplete, with obligate microbial symbionts providing additional micronutrients, table 6.1. Or an additional resource is provided, sufficiently valuable to reinforce mutualistic associations, such as sap-sucking insects excreting honeydew, which is utilized as an energy source by tending ants, table 6.1. Then there are protective mutualisms in which plants may provide a protected domicile, such as stems or thorns for ants to nest in, ants that protect sucking insects from natural enemies, or microbes that provide antibiotics for their larger partner, as in polytenoviruses PDVs, in ichneumoniid and braconid wasps, see Chapter 8. In this kind of classification, one mutualism may confer two kinds of benefit, as is often the case. For example, aphids provide a new resource for ants in the form of honeydew, and this constitutes an exploitative mutualism for ants, while at the same time ants protect aphids from attacks by predators and parasitoids, a protective mutualism for aphids. A third kind of mutualism involves the dispersal of propagules such as seeds and fruits, together termed diaspores, meaning units of dispersal, the transmission of pollen from one plant to another thereby providing pollination services, or the dispersal of mutualistic mites from one resource to another by larger insect associates, table 6.1. Such mutualisms can be called dispersal transmission mutualisms, or transportation mutualisms. The value of pollination of crops by native wild bees in the United States alone is estimated at over $3 billion annually, Lassie and Bond 2006 providing a glimpse of their probable ecosystem value in natural vegetation, see also Kremen and Ostfeld 2005. Another category could be added to Table 6.1, namely indirect mutualisms, or complex interactions involving mutualists, but this addition would have to cover an enormous range of phenomena. For example, mycorrhizal fungi have direct effects on the plant associate, but they may alter plant quality, which impacts herbivores and even higher trophic levels, e.g., Gange et al. 2003. We will discuss this subject in greater depth later in this chapter, and in Chapter 13 on multitrophic interactions. However, it is worth keeping in mind the inevitable consequences. Table 6.1 Examples of mutualistic associations involving insects, an ecological perspective. Services provided by partner. Services provided by insect taxon partner taxon references A. Exploitative mutualisms, new resources exploited by insects. 1. Nutritional value of food improved, improved. Nutrition of blood. Transport, inoculation, domicile. Sucking lice, bed bugs, tsetse flies, triatomines, hippobosids. Nycterebiids. Microbial species. Chapman 1998. Derbasula et al. 2003, Aksoy 2003. B. Digestion and nutrition of keratin, wool, feathers, skin transport, inoculation, domicile. Chewing lice, clothes moths, trogid beetles microbial species. An assumed association, Robinson 2004. C. Improved. Nutrition of plant sap. Transport, inoculation, domicile. Thrips, cicadelids, psyllids, coccids etc. Microbial species. Douglas 2003. Vega and Dowd 2005. D. Improved. Nutrition of seeds transport, domicile rice weevil microbes Nardin and Grenier 1991. E. Improved nutrition of fruits. Transport, inoculation tephritid flies bacteria Lozon 2003. 2. New resources made available, digestion of wood and cellulose. Transport, inoculation, domicile. Longhorns, ambrosia beetles, wood wasps, roaches, termites etc. Fungi, yeasts, bacteria. Martin 1987, 6 2003. Sue and Blackwell 2005. Harrington 2005. B. Digestion of plant parts. Domicile, culture, transport. Leaf-cutter ants, termites. Fungi Curry 2001, Anand and Boomsma. 2005. Schultz et al. 2005. C. Nectar and pollen transport of pollen. Pollination. Moths, butterflies, bees, ants. Plants Bentley and Elias 1983. Rico Gray and Oliveira 2007. D. Extra floral nectar. Protection of supplier. Ants, parasitoids plants Bentley and Elias 1983, Oliveira and Oliveira. Filio 1991. Table 6.1 Kant. Services provided by partner. Services provided by insect taxon partner taxon references. For example, of mutualistic interactions for other species coopering in the same habitat. For example, ants protect aphids against predators and parasitoids, while receiving honeydew as a reward, but the ants may also protect the plant from other herbivores such as leaf beetles and caterpillars, e.g., Lane and Nemella 1980, Oguchi 2005, Oguchi et al. 2007. Thus, there is a network of indirect links involving mutualists, food resources and non-feeding interactions, such as providing domiciles or protection, resulting in community structures that are recomplex and inadequately studied, Oguchi 2005, Oguchi et al. 2007. 6.3 Mutualism and the evolution of biodiversity. Mutualism is implicated in practically every major step in evolutionary history. Even among the bacteria, and before eukaryotic cells evolved, there were undoubtedly beneficial associations developed in stromatolites, bacterial crusts and other kinds of communities on soil, rock faces and aquatic surfaces. Stromatolites are many-layered microbial mats forming stable, sedimentary, boulder-like structures in marine environments. In these interactions one species provided services to another, such as plasmids, which provide mutualistic services to bacteria today, e.g., in the crown gall bacterium, and cooperation was no doubt frequent as it is today in biofilms, e.g., Webb et al. 2003, Brockhurst et al. 2006, Hansen et al. 2007. Once photosynthesizing protists evolved the air thought to have combined with fungi to produce green plants, figure 6.1, the fungal associate foraged for nutrients and water in the soil, while alga provided photosynthesis and protection against desiccation above ground. Moreover, the first fossil plants were associated with endophytic fungi, which are likely to have been precursors of mycorrhizas today. Thus plants evolved more as ecosystems rather than individual species because of thesymbiosis involving the eukaryotic cell, photosynthesizing cyanobacteria, fungi in the thallus of the plant and mycorrhizas adding to the nutrition of plants as they became larger, figure 6.1. 6.3.1 Herbivore radiations. Once plants had evolved and grown in stature, they formed the resources for herbivore radiations, Price 2002a. The initial problem was that animals and plants have very different structures and chemistries, e.g., C/N ratios making plants nutritionally poor resources for animals, see Chapter 4. The solution in many cases was for herbivores and detritivores to acquire mutualistic symbiotic bacteria, protists or fungi, pre-adapted over long periods of evolutionary time to digest plant parts and to synthesize nutrients deficient in plants or detritus. And this is the way in which the adaptive radiations of insect herbivores became so successful. Many insects feeding on nutritionally deficient plant sap are associated with mutualistic symbionts, up to 7 species in some cases, Buckner 1965, including leaf hoppers, aphids, tree hoppers and jumping plant lice, Moran and Telang 1998, Table 6.1. Many wood-associated herbivores depend on fungi and other symbionts for their ability to digest cellulose. Longhorn beetles, ambrosia beetles, leucanid and passilid beetles, cirricid wasps, termites and wood roaches, Table 6.1. Additional evolutionary innovations have been to use fungi as pathogens which reduce the vigor of trees, or to kill them, thus opening up breeding sites for bark beetles, Beaver 1989, Weber and Gibbs 1989, or to carry fungi from plant to plant in societomyid gall-inducing insects, with the fungal associate providing food for the developing fly larva, Bissett and Burkett 1988. In these ways plant-based communities have developed, which involve adaptive radiations of species assemblages, more like ecosystems rather than individual species, Price 2002a, Figure 6.2. Even large animals can exploit nutritionally inadequate green plants with the aid of fermenting bacteria and protozoa, such as ruminants as depicted in Figure 6.2, and those with cecal fermentation such as horses, rabbits and hares. 6.3.2 Carnivores with mutualists. But, in the building of food webs, mutualistic associations do not stop with the herbivores. Carnivores also depend on mutualistic microbes in many ways, Table 6.1. These include 1. Blood feeders such as sucking lice, bedbugs and tsetse flies, in which microbes provide supplemental nutrients for insects feeding on a relatively homogeneous resource. Also hippobosids, nycterebiids and triatomites like Rhodnius. Not included are fleas, mosquitoes and horseflies, whose larvae feed aquatically or on detritus on nutritionally more diverse resources. 2. Insects that feed on homogeneous or nutrient-poor substrates such as dead skin and keratin, as in horn and wool, which can be exploited probably only with the aid of mutualists, e.g., chewing lice, clothes moths. 3. Parasitic carnivores on insects which depend on microbial mutualisms to aid in overcoming the defenses of their hosts, as in parasitic wasps and virus particles, or in protecting a host from invading bacteria or fungi, as in nematodes with symbiotic bacteria on scarabaeid larvae, Figure 6.2. In addition to microbial symbionts, insects themselves may become mutualistic with carnivorous insects. For example, herbivore products, such as honeydew from aphids and tree hoppers, and sugary secretions from lycaenid caterpillars, also provide nutritional supplements to the generally carnivorous ants, Tables 6.1, 6.2, Stiersky and Eubanks 2007. It becomes apparent, albeit rather slowly because so many associations are subtle and cryptic, but nevertheless convincing, that mutualisms play vital roles in communities and ecosystem functions, and in the evolution of biodiversity, Cake et al. 2004, Eastwood et al. 2006. Many ecological and evolutionary opportunities and pathways have been opened up the association of one species with another, with each providing novel resources to the other. This is particularly evident when apprehending the larger picture, alt how delivered piecemeal in Table 6.1. This complexity results because food webs expand as opportunities provide mutualisms enable more associations, richer communities and more complex ecosystem functions. Parasitic. Wasp larva feeding on fungal lining. Of gall. Gall fly. Beetle breeding in dying tree blue stain fungus beetle in brood gallery ambrosia fungus bacteria and protozoa in rumen. Large artiodactyl herbivore. Bacteria. Nematode worm. Root feeding beetle 6.3.3 Atrophic level perspective. Taking a trophic-level approach, plants have mutualists such as ecto or endomycorrhizas, and other endophytic fungi e.g., Clay 1988, Arnold and Lewis 2005, any of which may influence the quality of food for insect herbivores, so effects cascade up to higher trophic levels where herbivores may be positively or negatively affected e.g., Gehring and Witham 2002, Ganj et al. 2003. Also, plant food is so poor relative to insects' nutritional requirements that mutualists generally provide essential enzymes or nutrients, or they concentrate or recycle nitrogen. Chapman 1998, his Table 4.3. Plants also providifloral nectar, pollen and extrafloral nectar. Table 6.1. The herbivores, in turn, provide food for higher trophic levels, such as honeydew for ants, wasps and parasitoids, and sugary secretions from lycaenid butterfly larvae attractive to ants. Secretions by lycaenid larvae are frequently referred to as honeydew. However, honeydew is liquid discharge from the anus of certain Hemiptera. Triplehorn and Johnson 2005, p. 786. Then the virtually ubiquitous ants affect other species in the system, attacking and removing many herbivores, while promoting the welfare of honeydew-excreting aphids, coccids and others. These kinds of interactions are present in many terrestrial ecosystems, but in the tropics additional linkages have evolved, with food bodies provided for ants by the plant genera Acacia, Cecropia, Macaranga, the Melastome family and others, and ants and termites practicing agriculture with fungus gardens. Table 6.1. Ants' nests in turn alter nutritional substrates for plants, and ants carry seeds into and into their nests, where germination and establishment is improved. Ants create fertilized pockets of substrate that the plants locate by making their seeds and fruits attractive to ants. BD 1985, page .77. Termites, with their own symbionts, process vast quantities of plant materials, changing soil quality and nutrient status. And so, these ecosystem processes keep revolving in communities, with mutualistic associations a key to integrating the many species and interactions involved. Table 6.2. Kant. Category of mutualism. And species examples of benefits and or costs references. See Lasius ants, Aphis. Aphids. Costs, longer development, later progeny production, fewer embryos etc. Stadler and Dixon 1998. Deep tropical ants, hemipterans ants benefit from plant and insect exudates. Davidson et al. 2003. Tropical ants, hemipterans variable benefits and costs because of variable densities of herbivores, coccids and pseudococcids. Lapola et al. 2005. Insects and fungi, IP's peony bark beetle, Ophiostoma fungus increased brood production, reduced colonization of trees. Copper et al. 2004. Forbia fly, epiclo fungus. Transport of spermatia, consumption of hyphae and ascospores. Parker and Bulkman 1991. 6.4 The origin of mutualisms. The evolution of mutualisms is not easy to study because the transition from two free-feeding species, that is individuals feeding alone without the aid of mutualists, into a mutualistic association is likely to be rapid when the selective advantages to both are great. However, we can speculate that antagonistic relationships such as competition AA, or parasitism A, are likely to be ameliorated by natural selection. Figure 6.3. In particular, parasites, already in a symbiotic relationship with their hosts, may benefit from the evolution of less pathogenic impact, with movement towards commensalisms, and ultimately mutualism. Price 1984b. Competitors are probably less likely to become mutualists because they utilize the same resource, so aiding each other is problematic. Mutualisms usually develop between organisms utilizing different resources. They are often on different trophic levels, and are taxonomically divergent, as illustrated in figures 6.1 and 6.2. Although we do not have an insect example of the observed transition from a parasitic symbiont to a mutualist, in a brief sequence of events, there is such an example between a protozoan and a bacterium. Plus, only 10 years elapsed before an amoeba culture became fully reliant on a formerly parasitic bacterium, with cloning becoming impossible in the absence of the bacterium, e.g., Jaon 1972, Jaon and On 1978. In another case, a parasitic plasmid became a mutualist with a bacterium, Escherichia, in a short period of 50 generations, Boma and Lenski 1988. The time periods involved in the transition from parasitism to mutualism in these cases are as a blink of an eye in relation to the evolutionary history of life in general, and such evolutionary opportunism and saltational adaptation is no doubt continuing unobserved in multitudes of cases today. Many examples have been noted in the literature where the shift from parasitism to mutualism has been suggested, such as mycorrhizas, Harley 1969, other endophytic fungi, Clay 1988, and herbivorous parasites, Westerberg 2004. 6.5 The give and take in mutualism. Very little is free in nature, there is no free lunch. If an individual gains in fitness, there is usually an associated cost. In the case of mutualism, this is particularly clear because of the mutual and reciprocal give and take involved. 6.5.1 Plants, pollinators and seed predators. An example of this gain and loss concerns the Sonida cactus, Lithosarius shati, which is pollinated by the Sonida moth, Upiga virescens pyralidae, in the Sonoran Desert. Fleming and Holland 1998, Fleming 2002. The female moths collect pollen from the cactus on specialized abdominal scales and deliberately deposit pollen on the receptive stigma of the flower. Then the female lays a single egg on a petal. Larvae bore into the ovary and eat young seeds for 6 days, then they leave the ovary, bore into cactus stems and emerge as adults. The cactus is an obligate outcrosser, with reduced nectar production and a limited ability to attract other pollinators. The cactus receives very effective pollination, 75% of fruits set in 1995, and 90% in 1996, but at a cost of larval destruction of about 30% of the seeds produced from pollination by Sonida moths. Although all seeds are consumed by a larva in an ovary, moths evidently pollinate several other flowers without laying eggs. However, many pollinated flowers are aborted resulting from limitation of resources such as water. Consequently, mortality of larvae may be an important regulating factor in the interaction. Holland et al. 2002. For the energetic cost of pollinating flowers, the moth receives a reward of a nutritious food resource for its larva. There is a net gain in fitness for each species because, in the absence of moths, pollination would be much less effective by default pollinators such as halictid bees, and net seed production would be lower. Thus, without providing the service of pollination, the pyralid would experience a much lower carrying capacity of ovules as larval food. Both cactus and pyralid populations would probably decline in the absence of the mutualism. This mutualism has been modeled by Holland et al. 2002 using net functional responses of pollinator and flower number, incorporating costs and benefits to the interacting species. The results suggest that fruit abortion may enable equilibrium in the mutualism. Features of the Sonida and Sonida moth system are similar to the yucca and yucca moth relationship studied by Pelmer et al. 1996a, b, as pointed out by Fleming and Holland, 1998. 1. Resource-limited fruit set is apparent in both systems, with the limiting resource likely to be water. 2. Flowers are obligately outcrossing and little nectar is produced. 3. Flowers are short-lived and open nocturnally. 4. Female moths collect pollen with specialized structures and deliberately transfer it to stigmatic surfaces of the flower. 5. Alternative pollinators are unreliable or absent. 6. Larvae destroy about 20–30% of seeds in a crop and yield a benefit, cost ratio from 2 to 5. More recently, Fleming 2002 estimated that the benefit, cost ratio was about 3:1 in the Sonida mutualism, a ratio similar to that seen in other pollination mutualisms of yuccas and their moths and figs and their fig wasps. We have mentioned 3 cases of more or less obligate pollinators whose larvae consume developing seeds, the Sonida cactus, yuccas and figs. We will expand on the yuccas and figs later in this section. A 4th case concerns the globe flower, Trollius europaeus ranunculacei, and 4 fly species in the genus Chiastocheta, Diptera, Anthemiidae Pelmer 1989. The flies are thought to be the exclusive pollinators of these globe-like flowers close to other potentially pollinating species such as bees. The flies mate, feed on pollen and oviposit within the flower, and larvae eat a few seeds each. As in the other pollinator and seed-feeding mutualisms, seeds are the currency in which the benefits and the costs of larval feeding are measured. Hence, it is relatively simple to estimate benefits and costs, which explains why these systems, although uncommon in nature, have been featured in the study of mutualism. As the number of flies increases per flower, pollination increases so the seed initiation frequency increases, figure 6.4. Also, the number of eggs laid per flower may increase, meaning that more seeds are consumed by fly larvae. However, neither trend is linear and relative seed set remains remarkably stable, around 36% over the range of egg densities per flower observed in nature, from about 2 to 7 eggs per flower, see also Jaeger et al. 2000. At the peak of relative seed set, with 4 eggs per flower, pollination services yielded a benefit of about 62% of ovules originally available in the ovary, and larval seed predation inflicted a cost of about 14% seed loss, leaving a net benefit of about 48% of seeds. This ratio of benefit to loss is therefore about 3.41, comparable to estimates mentioned above for other systems. Further studies have found that the interaction of host plant and pollinators, seed predators was always beneficial for the plant in 20 natural populations in France, Jaeger et al. 2001. Of course, both species in a mutualism experience costs, so selective conflicts emerge, with each species maximizing gains and minimizing losses. Such conflict was emphasized by Bronstein, 1992, see also Anstead et al. 1996, in the fig and fig wasp interactions, where she examined in detail how the evolutionary balance between participants could persist for so long. The pollinating wasp would appear to have an evolutionary advantage, passing through about 100 generations per single fig tree generation. So, why doesn't the fig wasp evolve with a longer ovipositor, enabling it to reach and utilize more ovules as larval food? What countermeasures have evolved in the fig? Janssen, 1983, page 232, even called mutualisms, reciprocal parasitisms, because the negative effects of partners were more or less balanced. Let's examine the fig and fig wasp. 100. Relationship in more detail as it represents a classic case of mutualism. Fig trees and fig wasps, from Price 1984b, 1997. More than 900 species of figs ficus, morrisii, have been recorded, which are pollinated by very specific wasps in the family Agonidae chalcidoidea, the fig wasps. Many characters in both the figs and wasps show closely coevolved properties, and the two taxa have obviously radiated together as a result of the initial development of a mutualistic relationship, Bronstein and McKee 1989, Gelberg et al. 2005. The fig is a false fruit formed by the enlarged receptacle of the inflorescence. The flask-shaped fruit encloses a large number of flowers, figure 6.5, and each inflorescence passes through the following stages: Galil and Isakowicz 1968, Ramirez 1970, Wiebes 1979. Phase of pre-male, in which the inflorescence is closed to entry by fig wasps, figure 6.6. Phase B, female, in which the ostiolar scales loosen, female flowers ripen and agonid wasp females penetrate the inflorescence and oviposit into the ovaries. Phase C, interfloral, where wasp larvae develop in developing galls formed from the fig ovaries. and unattacked fig embryos develop. Phase D, male, where male flowers mature, wasps reach maturity and emerge from galls. The males inseminate females and bore holes in the receptacle. The females collect pollen from the male flowers and emerge through the holes bored by males and fly to fig inflorescences in Phase B, and Phase E, postfloral, in which seeds ripen and the receptacle ripens, becoming attractive to fruit-eating animals, which disperse the seeds. The life cycle of the fig wasps is also complicated, and only a general pattern is depicted here, as there is considerable variation between species. Before females leave the inflorescence in Phase D, they load up with pollen, packing it into special receptacles on or near the coccyx of the front legs and the abdomen. Then they exit from the inflorescence through holes bored by males, fly to figs in Phase B on another tree, and enter the ostiole. On entry they lose their wings and part of their antennae. Males are wingless, and do not leave the fig fruit in which they developed. In the lumen of the fig, females pierce with the long ovipositor the stigmas of the flowers and the length of the styles, ovipositing in the ovary, figure 6.7. After ovipositing in an ovary they pollinate the flower by scraping pollen out of the receptacle with their legs onto the stigmatic surface. They also pollinate flowers in which no wasp eggs are laid. Eggs hatch and larvae develop in the gall formed from ovary tissue. Males emerge before females, cut holes in the sides of ovaries, and inseminate the female inside the gall. Females then collect pollen and leave the fig, completing the life cycle. Some coevolved traits in the figs include 1. The unique false fruit design, allowing only agonids and a small number of closely related parasitic wasps to enter the inflorescence. Bored by malamale flower ostiole male. Flower. Figure 6.5 diagrammatic crop. 2. The extreme protogeny, with female flowers receptive several weeks before male flowers. 2. The extreme protogeny, with female flowers receptive several weeks before male flowers produce pollen, is clearly adapted specifically to the generation time of the fig wasp. 3. The inflorescence contains both stalked and unstocked flowers with short and long styles, respectively, making seeds more or less available to ovipositing fig wasps, some sacrificed for the mutualistic wasps and some reserved for seed production. Some coevolved features of the wasps include 1. The specialized morphology of both male and female fig wasps, figure 6.8, involving a. The female body adapted to squeezing through the ostiole of the fig, b. Pollen receptacles on the female, c. A wingless male with a long abdomen for mating with a female in the gall. 2. The specialized behaviors of loading and releasing pollen. 3. The specialized secretions in the female that promote gall formation. 4. The very specialized relationship, usually between one fig species and one wasp species, reminiscent of the highly specialized parasites discussed in Chapter 8. Of course, with so many fig and fig wasp species, few have been studied in detail so that further explorations will yield new and fascinating information. But we now understand that the costs and benefits of this relationship are extensive and multifaceted. 6.5.2 Ants and plants. Since the first benefit and cost analysis of pollinator and seed predator by Pelmer, 1989, these and many other systems have been examined. Table 6.2. Ants benefit plants, and the plants provide food for ants in the form of extrafloral nectar and ants benefit plants, and the plants provide food for ants in the form of extrafloral nectar and A, B, C, E. Protein-rich food. These many ant plants, or myrmecophytes, also provide shelter in the form of swollen plant parts, as in Bull's hornacaceous, Janssen 1966, 1967a, b, or hollow stems as in Cecropia species Davidson et al., 1991, into which the ants can bore and establish their nests. We will consider the C examples in a little more detail as they have become classic examples of mutualism, as in the case of figs and fig wasps. Bull's hornacacia and Cecropia species and ants partly from Price 1984b, 1997. More than 90% of species of the genus Acacia Fabaceae, Mimosoideae, in Central America are protected from herbivores by cyanogenic chemicals in the leaves. Rare et al. 1973. The remainder seem to have gained a more potent defense in the form of ants which live in close association with these plants. Belt 1874 discovered that some species, the Bull's hornacaceous e.g., Acornigera, act as hosts to colonies of ants in the genus Pseudomyrmex, figure 6.9, and the ants act as allelopathic agents. Fourth plant. Brown 1960, Janssen 1966, 1967a, b. The ants gain protection from the plant by living in the swollen stipular thorns, and food is provided by the plant. Sugar is secreted by petiolar nectaries and protein is produced in small beltian bodies growing at tips of new leaflets. Figure 6.10. The aggressive ants patrol the plant, ward off herbivores and suppress potentially competitive plants by chewing growing tips. Janssen 1967b. Such suppression of plants around an occupied acacia plant also makes it much less vulnerable to fire, which frequently sweeps through this dry tropics vegetation. Janssen 1967a. Similar relationships exist between ants and Scropiaceae plants. Figure 6.11, Janssen 1969, Davidson and Fisher 1991, Davidson et al. 1991. Ho diaresis LL Dobler and Wilson 1990, in which the precision of coevolution can be seen in the production of animal sugar, glycogen, in the mu diaresis Aryan bodies of the host plant, the only noun case in the higher plants, Rickson 1971, 1976, see also Buckley 1982. About 100 species of Scropi in the New World tropics house ants in their holostems. The plants provide prostomata which are resimal areas in the internodes through which queen ants can easily cut to gain entrance to the holostem, which becomes a brood chamber. After entry, the prostoma grows back to close the opening, protecting the queen as she lays her eggs and rears her first brood of workers. Workers open the prostomogon and forage for mu diaresis Aryan bodies rich in glycogen produced in Trichilia platforms at the base of petioles, figure 6.12. Other food types Provididae. Various Cecropia species are spongy parenchyma cells on the stem inner walls, pearl bodius rich in lipids on leaf surfaces, and honeydew produce coccids tended by the ants in the hollow stems. Davidson et al. 1991. Figure 6.7. Female fig wasp, Ceratosole arabicus, ovipositing in a short-styled flower of Ficus sycamoris, and extracting pollen from a pouch to fertilize the flower. From Galil and Isakowicz, 1969. Other well-studied ant and plant interactions include the plant genera Leonardoxa, Caesalpiniaceae, and Macaranga, Euphorbiaceae, Hyland McKee 2003, and references therein, which would be rewarding for the reader to explore. The full array of ant and plant associations is covered by Beattie, 1985, Huxley and Cutler, 1991, Ho diaresis LL Dobler and Wilson, 1990, Enrico Gray and Oliveira, 2007. Ant dwellings, or domatia, also Myrmecodomatia, may also house mutualistic hemipterans such as coccids and pseudococcids, from which the figure 6.8 inhabitants of Ficus sycamoris inflorescences. The female Ceratosole arabicus, Agonidae, is the pollinator and mutualist. Psychophaga sycamori, Agonidae, is a parasite of the fig and a competitor with C. arabicus because it does not pollinate the fig, but utilizes both long and short-styled flowers as oviposition sites. Apocrypta longitarsis, Torimidi, is an inquiline utilizing galls formed by Ceratosole and Psychophaga. From Galil and Isakowicz, 1968. Ants derive carbohydrate resources in the form of honeydew. A case is provided in Table 6.2 under ants and hemipterans, Lapola et al. 2005. When ants feed one honeydew produced by a hemipteran, for example, the process is called trophobiosis and the provider is a trophobiont. Such cohabitation with ants is also called myrmecophily, which is practiced by myrmecophiles, although this term was originally reserved for the ants, guests, in the nest. Wheeler 1910. Hence, all hemipterans excreting honeydew and lycinids secreting sugary liquids were regarded as trophobionts, but only those living in an ant's nest were considered to be myrmecophiles. However, this definition still includes many aphids, coccids and pseudococcids that reside in ants' nests, e.g., Lapola et al. 2005, and lycinid larvae that. Figure 6.9. The ant, Pseudomyrmex, with a long, thin, body form suitable for living in hollow twigs, stems and thorns. From Ho diaresis LL Dobler and Wilson 1990. Figure 6.10 right. The end of a branch of a bull's horn acacia, Acacia spherocephala, with paired, inflated and hollow thorns inhabited by ants in the genus Pseudomyrmex. Entry holes cut by ants are marked with an X. Left. A young shoot of the same acacia species, with beltian bodies at the tips of leaflets, enlarged in inset at lower left, and an extrafloral nectary on the upper surface of the petiole, marked by AY. The young thorns have not been occupied by ants. From Wheeler, 1910. Enter nests of ants, e.g., Thomas 1980, 1989, 1991. Enter nests of ants, e.g., Thomas 1980, 1989, 1991. At present, the term myrmecophile is defined as an organism that must spend at least part of its life cycle with ant colonies. Ho diaresis LL Dobler and Wilson 1990, page 640, which would presumably exclude facultative trophobionts. One case study concerning ants and lycinid larvae shows how variable relationships are. With this context dependency, Bronstein et al. 2006 are conditional mutualism, Cushman and Witham 1989, Cushman and Adicott 1991, Bronstein 1994, responses of larvae dependent on ant density and ant species, figure 6.13. Figure 6.11 Myrmecophytic Cecropia tesmani, showing a swollen stem with an open prostoma, an entry site for ants, and petiolar bases with white trichilia, sites of mu diaresis Aryan body production. Ants are workers of Pachychandila luteola. The new name for the plant is provisionally Cecropia pungara, with the specific name relating to the stinging ant named by the local people. Figure by Ali Partridge based on photograph by Diane Davidson. From Davidson et al. 1991. A. B. Figure 6.12 details from the myrmecophyte, Cecropia adenopus, which grows in the American tropics, and supports ant colonies in the hollow internodes. Note the pads at the base of the petioles which produce mu diaresis Aryan bodies fed upon by the ants. B. An enlarged surface of a petiolar base shows developing egg-shaped mu diaresis Aryan bodies. From Ho diaresis LL Dobler and Wilson, 1990. Reprinted by permission of the publisher from the Ants by Bert Ho diaresis LL Dobler and Edward O. Wilson, Cambridge, Massachusetts. The Belknap Press of Harvard University Press, p. 222. Copyright #1990 by Bert Ho diaresis LL Dobler and Edward O. Wilson. In all cases in Table 6.2, and in the literature in general on ant and plant mutualisms, there is an emphasis on benefits and costs to the plant, reduced herbivora, increased fruit and seed set, with the cost of providing extrafloral nectar. Seldom are the ants the focus of a benefit-cost analysis, but see Pierce 1989 discussed below. Moving on from ant relationships, we list in Table 6.2b cases where pollinators and seed predators have been evaluated in terms of benefits and costs in the relationship. This kind of relationship has been named brood site pollination mutualism, Sakai 2002, and nursery pollination, Dufay diaresis and Anstedt 2003. Again, emphasis has been on the 6543210.51.01.52.02.53.03.54.05. Tapinoma sessile. Formica. Altipedans. Formica obscuripes. Dropl at S10. M. In. Mean no. Of attending ants. A. 80. 40. 20. 124 treatment. E versus ERSI on S10. M. In. 8. 60. Formica. Obscure. Figure 6.13 variation in the response of lycaenid larvae, Glaucopsyche ligdanus, to different species of tending ants, Formica altipedans, Formica obscuripes and Tapinoma sessile. A. Droplets of sugary secretions produced by ant-tended lycaenid larvae increased as the number of ants touching and tending larvae increased, particularly when T. sessile was tending. B. Also, tentacle aversions decreased as the number of ants tending increased in T. sessile, the treatment axis. Aversible tentacles in lycaenid larvae are thought to release volatiles, which alert ants and increase attendance. Modified from Axon 2000. Plants gains and losses in the interaction. However, the example of Pelmer 1997 actually measured costs to the yucca moth. These were very low. Time allocated to pollination, 4.1%. Specialized morphology, 0.42% of female body mass. Pollen load transport, less than 4% of female body mass. Pelmer concluded that with such modest costs, one would expect the pollination, seed predation syndrome to be more common than is observed so far in nature. Perhaps many more cases await discovery, or the copollinator abundance hypothesis, Pelmer 1997, page 1658, may limit evolutionary opportunities. For example, if copollinators are effective on a plant, then specializing on that plant will be unrewarding because pollinators would not limit seed set. And, indeed, the range of nursery pollination systems noted in the literature has expanded in the last decade, e.g., Sakai 2002, Dufay diaresis and Anstedt 2003. 6.5.3 Ants and herbivores. Ants and lycaenid larvae, and ants and hemipterans show similar relationships, with ants providing protection against predators and parasitoids, and gaining sugary secretions or honeydew, table 6.2c. Much of the literature assesses benefits and costs to the herbivorous insect rather than the ant partner. Frequently, the costs of being tended by ants and supplying nutrients appear to be high, as evidenced by reduced pupil and adult weights decreased by 25% in lycaenids, and smaller body size, fewer embryos, longer development times and delayed progeny production, all of which have been recorded in aphids and other hemipterans, table 6.2. Moreover, some species of aphids, coccids, membracids and lycaenids have become obligate associates of ants and are unable to survive in the absence of ants, see Stadler et al. 2001 for a good review of costs and benefits of mutualistic interactions with ants. Pierce 1989 assessed benefits to ants tending lycaenid larvae by measuring weights of ants ascending trees to tend larvae, and weights as they descended. The increased weights of descending ants documented a significant energetic gain as a consequence of the resources garnered by tending lycaenid larvae. The metabolic cost of foraging was at least compensated for in the tree canopy because of the net gain in weight off ants, but the extent to which such foraging benefited the ant colony at large, and the queen, was not determined. 6.5.4 Insects and fungi. The last category of mutualism treated in Table 6.2 is the associations of insects with fungi. Costs and benefits have not been analyzed frequently in this literature, although it is extensive. See Batra and Batra 1967, Batra 1979, Martin 1987, Pirozinski and Hawksworth 1988, Wilding et al. 1989. Specialized pouches for transmission of fungal spores or mycelium, mycangia or mycetangia, from one host plant to another have evolved repeatedly, being present in wood wasps, bark and ambrosia beetles, Cystidomia gal-inducing midges, leaf-cutting ants and others. These fungus-bearing pouches are invaginations off the epidermis with external openings, as opposed to internal mycetomes, bodies of shells containing symbiotic organisms within host organisms, Buckner 1965. In an example involving the bark beetle, Ipes pini, and the blue stain fungus, Ophiostoma, Copper et al. 2004 introduced beetles into the cambium of pine trees which had been inoculated with fungi at various times, 0, 3, 7 and 10 days before beetle entry to determine the effects on bark beetle colonization and development. As the blue stain fungus developed longer, colonization rates declined, but the number of progeny increased relative to fungus-free controls. Since attack of dying trees and inoculation of blue stain fungus by beetles is normalis simultaneous, because the fungus is carried by the beetle in mycangia, the mutualistic association benefits the beetle both because more progeny are produced by the colonizing cohort, and because competition from colonizing conspecifics is deterred. Of course, the fungus benefits from highly specific transport from host to host and effective inoculation into the cambium of the host. Another insect-fungus mutualism involves the Forbia fly and the fungus epiclo. Epiclo is an endophytic ascomycete on the grass, Elamus virginicus, table 6.2. This fungus forms stroma on the grass stem surface in the spring, producing spore-like spermatia. The flies are attracted to the stroma and carry spermatia to another stroma of the opposite mating type, thus acting as the equivalent of a pollinator. The flies oviposit on the stroma and larvae feed on fungal tissues, including ascospores, which develop after the fungal mating types fuse. This example is therefore equivalent to the mutualistic examples of pollinators and seed predators. There are many interesting questions which remain to be addressed regarding mutualistic associations. What is the variation in the intensity of the mutualistic relationship over the landscape? For example, when different ant species tend lycaenid larvae, e.g., Axon 2000, or when extrafloral nectar traits vary, Rogers and Strauss 2004. And why don't extrafloral nectaries result in protection of the plant against herbivore attack in some cases, e.g., Rashbrook et al. 1992. Also, at least 3 species are usually involved in a mutualism because the association is often involved with protection against natural enemies, so variation in any one of the players is important and interesting consequences. Fourth, strength of the interactions, see review by Bronstein and Barbosa 2002. In ant and plant interactions, hemipterans may become involved, and the species may change with time or across habitats, posing the question of how this alters the ant's protection off the host plant, e.g., Gum et al. 1998. More perplexing and esoteric questions arise in the fig and fig wasp interactions because some fig wasp species cannot reproduce in figs with only female flowers, begging the question as to why they enter such figs only to pollinate, e.g., Moore et al. 2003. It is in the nature of mutualism that interactions are complex and highly variable, Bronstein 1998, with geographic mosaics of coevolution common, Thompson 2005, see Chapter 4. The study of benefits and costs of these interactions force detailed analyses and raise many questions, ensuring a rich future for their continued study. 6.6 Cheating. Where there are rewards, cheaters will materialize, both in human society and in mutualistic systems in the wild. If a pollinator's activity results in an ovary full of seeds, why not use the seeds for larvae without the bother of pollination? 6.6.1 Seed predators. Cheaters in the yucca moth and yucca mutualism have evolved at least twice, figure 6.14, Pelmer et al., 1996a. The cheaters have evolved to be finely tuned Toyuca species phenology. Following pollination in Yucca filamentosa, fruits with more than 7 or 8 myth eggs are abscised within 5 days, a response to the inevitable overexploitation of seeds by so many resulting larvae. Fruits that are not abscised will normally have 26% of seeds consumed by larvae from the pollinating moth. However, the early phenology cheaters oviposit into the fruit after all abscission has been initiated, and consequently do not influence the probability of fruit loss. Thus, cheaters feed on and destroy more seeds than the pollinators, consuming from a mean of 75% of seeds up to 100% in some fruits. Late phenology cheater species oviposit 2 to 3 weeks after pollination into the fully grown fruit, using a long ovipositor. When both types of cheater species are present, almost all seeds are destroyed. The phylogenetic hypothesis on tegaticula moths suggests multiple host shifts among yucca species, opening up opportunities for one species of pollinator to move to a new host and remain a mutualistic pollinator, while another with a slightly delayed oviposition time could evolve into a cheater. It would appear that the pollinator strategy is more rewarding, in terms of larval survival, than the cheater strategy, or if long-term coexistence of the association would be impossible. Since the genus Tegaticula evolved, cheatershave coexisted with pollinators for about 35% of the lineage's existence, a very long persistence time for this potentially destabilizing set of interactions. 6.6.2 Flower visitors. Another form of cheating occurs when nectar is taken from flowers without pollination. Nectar robbing, e.g., Inoue 1983, Scott et al. 1993. This is known to occur in birds, bees and ants, where for example, large carpenter bees in the genus Xylocopa pierce the base of the corolla of tubular Solanum andocodio, Fucaria splendens flowers, and suck up nectar without contacting stamens or stigma at the opening of the flower. Xylocopa bees are short-tongued and rob many plant species, and together with short-tongued bumblebees Bombus spp. And stingless bees Trigona spp. Hundreds of plant species are robbed. These bees form a class of 100, 100, 100, 100, 83, 36, 9,975 88, 87, 73, 3,131 58, 100, 88, 78, 100. Late cheaters. Early cheaters. M. ESE Pi OLA. SPECCA. Y. WHIPPLEIS. CA. Y. WHIPPLEIN. CA. Y. BREVFOLIA. NB. Y. TORREYITX. Y. Primary robbers, which cut into flowers, but this provides access to nectar for secondary robbers whose mouthparts can enter slits at the base of corollas already present. Honeybees, moths and butterflies may all act as secondary cheaters. The costs of nectar robbing to the seed set in flowers appears to be minor, because normal pollination by long-tongued bees, butterflies and moths is not curtailed, Morris 1995. In some cases seed set may be increased by cheaters because reduced nectar supplies results in pollinators visiting more flowers. However, although nectar robbers may range from cheaters to mutualists, Malouf and Inoue 2000, generally they have a detrimental effect on plant reproductive success, Irwin et al. 2001, although this effect is highly variable in time and space, Irwin and Malouf 2002. Cheating is not restricted to the insect and bird pollinators because some plants cheat in the interaction as well. For instance, deceit pollination is observed in orchids, begonias and other plant species. Fritz and Nielsen 1996, Schemsk et al. 1996. In orchids, such as in the genera Ophrys, Cullenberg 1961, and Orchis, Fritz and Nielsen 1996, flowers provide no apparent reward to pollinating bees, but resemble females of the pollinating species both visually and pheromonally. Males attempt to mate with the flower, and carry away pollinia after flower visitation. This is called pseudocopulation. Similar deceitful relationships have evolved in plants with flower parts mimicking the hosts of parasitoids, attracting female parasitoids which sting into the flower and pollinate it. Pseudoparasitism, Faegre and van der Pijl 1971. Flowers may also mimic other insects, causing bees to attack in territorial defense, which results in pollination via a deceptive process called pseudoantagonism, or pseudoaggression. Another form of deceit pollination occurs when flowers are unisexual, and female flowers offer no reward to pollinators while males provide pollen, as in the genus Begonia, Schemsk et al. 1996. Although male flowers are larger, displayed more conspicuously, and offer a reward, female flowers attract brief visits which result in pollination, by mimicking male flowers. Spectral qualities are similar. UV light absorption is strong by anthers and stigmas, and sepals are similarly reflective in the UV rangine male and female flowers. Both male and female flowers are formed, mainly by two conspicuous sepals, and the sex ratio is female biased, about 66% females. Although male flowers were much more attractive to bees than females, experiments with supplemental pollinations produced no more fruits per plant than controls, which received natural pollination, and in both treatment and control about 45% of female flowers produced mature fruits. However, with supplemental pollination, seed set was higher by about 10%. Nevertheless, with little cost to male flowers, except that of looking like males, which is usually avoided in nature, this deceit pollination through intersexual mimicry is effective, evolutionarily stable and of long standing. Many other forms of cheating occur in mutualistic systems, because the opportunities for cheating are almost limitless. Lycaenid butterfly larvae drink extrafloral nectar provided by the host plants to attract ants involved in protecting both the plant and the caterpillar from natural enemies. DeVries and Baker 1989. Non-pollinating wasps utilize fig fruits pollinated by others. Bronstein 1991, and parasites of mutualisms are commonplace. U 2001. Inquilinism is the relationship in which a species spends much of its life cycle in the nest of its host species. In fungus-growing nests of ants and termites, and other myrmecophily without rewards for ants, inquilinism is represented by hundreds of species. Ho Diaresis LL Dobler and Wilson 1990. Nonetheless, the advantages of mutualisms clearly outweigh the drawbacks incurred from conflicts of interest and cheating. However, some clades ancestrally mutualistic show defectors in which species become parasitic or independent. Saxon Sims 2006, as with the case of cheating yucca moth species. 6.7 Adaptive radiation in mutualistic systems. Many authors have argued for the importance of dead organic matter as a cradle of evolution for insects. See Hamilton 1978. These kinds of habitats would bring the colonizers into intimate contact with the multitude of microorganisms already adapted for living in and on wood, with the necessary enzymes for digesting organic matter, and toxins for dealing with antagonistic interactions. Acquisition by insects of such organisms, as mutualists, would proceed more rapidly than in the de novo evolution of the necessary traits, and result in more efficient exploitation of resources. We provide some examples, starting with radiation in woody substrates, moving to roaches and termite symbionts, and then we consider the role of symbiosis in the radiation of the ants. 6.7.1 Breeding structure in dead trees. Hamilton narrowed the focus to dead trees in his article, Evolution and Diversity Under Bark. He emphasized the evolution of breeding structures that resulted in rapid evolution and adaptive radiation. 1. The origin of wing polymorphisms is frequent, as observed in Zoroptera, Socoptera, Thysanoptera, Hemiptera, Coleoptera, Diptera and Hymenoptera. Food sources under bark for these groups were fungi, yeasts and small arthropods. 2. The origin of male haploidy occurred at least 5 times in this habitat, Hymenoptera, Thysanoptera, Micromalthus beetles and some scalidid beetles, as well as some mites. 3. Eusociality has evolved at least twice, in the termites and ants, and possibly in the social aculate wasps. 4. Sexual dimorphism is also common in wood-dwelling insects. Think of the stag beetles, the Lucanidae, with large males with huge mandibles like antlers for fighting, and the smaller females without enlarged mandibles, figure 2.11. Several orders have species with winged males and flightless females, Dictyoptera, Embioptera, Socoptera, Hemiptera and Coleoptera. The breeding structure resulting from these 4 common attributes of insects in wood results in small interbreeding units in which natural selection and drift may be rapid. Many such isolated demes would indeed create a cradle for evolutionary innovation, and many major groups appear to have originated in wood. Dictyoptera, Isoptera, Zoroptera, Dermaptera, Thysanoptera, Hymenoptera, Symphyta like cirricid woodwasps, and Coleoptera. In these small interbreeding groups, exchange of beneficial microbial associates would be effective and probably common. Bacteria, protozoa, yeasts and fungi would also be common, yielding plentiful opportunities for symbiotic associations. Wood is difficult to digest, and cellulases, which are not produced by most insect groups, are most commonly found in microbial species and fungi. Table 6.1. Also, wood low in nitrogen provides inadequate nutrition for insects which are contrastingly high in nitrogen and protein. However, this depauperate substrate can be utilized by microbial and fungal partners from whom the insects benefit. Although Hamilton, 1978, emphasized breeding structure, he did mention symbiosis as one factor that may facilitate evolutionary change, but the case can be made that symbiosis was perhaps at least as important, if not more so, than breeding structure. This results because, although breeding structure has changed during radiations, symbiosis has been maintained throughout radiations in many cases, suggesting its more central role in the process. We will examine 3 examples of adaptive radiation of major insect taxa in support of this contention: the Dictyoptera, the Scarabaeoidea, and the ants. 6.7.2 Microbes in cockroaches and termites. The Dictyoptera include the termites Isoptera, mantids, Mantodia, and the cockroaches Blatodia, Triplehorn and Johnson 2005, Gullin and Cranston 2005. The mantids are predatory, so symbiotic microbes are not utilized, but in the termites and cockroaches mutualistic microbes have been basic and central to the adaptive radiation of these 2 orders. Nalipa et al. 2001, p. 194. We suggest that the evolution of a sophisticated hindgut fauna is a process of internalizing this self-assembled microbial community, accompanied by changes in host microbe interdependence, the source of microbial inoculum for neonates, and host social behavior, and the association of these flagellates with the Dictyopteran lineageis and ancient one, and may have originated as part of the external rumen in the Carboniferous coal swamps, i.e., over 300 million years ago. The external rumen mentioned here refers to feces in which microbes act as potential mutualists with the precursors of termites and roaches, providing breakdown of cellulose, detoxification of chemicals, a softening of the substrate, and a direct source of nitrogen-rich food. Thus, digestion is partially external to the insects, but equivalent to the microbial activity in the rumen of ungulates. It is argued that the ancestors of the Dictyoptera fed mostly on rotting plant material in which fungi, protozoa, and bacteria would be active in decomposition, so associations of microbes and insects were inevitable. After ingestion of an active culture of plant material and microbes, decomposition may well accelerate, and anaerobic fermentation became possible. Some microbes would be retained in the gut and eventually become accommodated in an enlarged paunch, the fermentation chamber in termites. Fecal pellets would provide a rich source of predigested plant material plus microbial protein and potential symbionts, so coprophagy, the consumption of feces, would be highly advantageous. Figure 6.15. Then, association among individuals in small family groups, as envisioned by Hamilton, 1978, would promote the coprophagous habit, and eventually highly social units and eusociality. Direct transfer of fecal material among family members, from the anus of one to the mouth of another, or proctodel tropholexis in cocktail party parlance, would ensure inoculation of neonates with mutualistic microbes, including groups such as flagellated protists which function in a liquid medium. Such vertical transmission of mutualists from parents to progeny would accelerate coevolutionary adaptation in the host and its gut fauna, and among faunal components themselves. Thus, symbiosis and mutualism appear to be fundamental to the evolution and adaptive radiation of the cockroaches and termites. 6.7.3 The Scarabaeoidea. Not surprisingly, another adaptive radiation originated from the detritivore habit, that of the microbes refractory food item. Metabolites, exoenzymes. B, F, E, A, E, E, C. Superfamily Scarabaeoidea, which includes stag beetles, bess beetles, rain beetles, dung beetles, skin beetles, scarab beetles, white grubs, and several families without common names. Table 6.3. But some members are greatly admired and even revered, like the sacred scarab of ancient Egypt, Hercules beetles, the Goliath beetle with larvae weighing almost 100 grams, Acorn 2007, and some large stag beetles with mandibles of prodigious proportions. The group has been radiating for about 200 million years, from Triassic times, and the existence of Pangea. The ancestral scarabs appear to have fed on humus as larvae, and fungus as adults. Figure 6.16, Schultz and Chown 1995. Present-day larvae have an enlarged hindgut which acts as a fermentation chamber. Figure 6.17. This gut morphology must have evolved early in the lineage because the trophic link to humus, dung and rotting wood, necessitating microbial digestion, i.e., fermentation, has remained as a theme throughout the radiation. Figure 6.16, Table 6.3. The fermentation of refractive substrates opened up huge opportunities to exploit a wide range of previously unavailable foods, as listed in Figure 6.16 and Table 6.3. Larvae fed within the medium, often boring into the ground, a hypogeal or below-ground level habitat, and adults would burrow into a substrate to oviposit. Hence, the colonization of wood, dead animals, and the nests of ants and termites would result from an instinct to burrow. In the scarab radiation, one clade expanded after the Triassic-Jurassic extinctions at about 208 million years ago, the Mesozoic clade shown in Table 6.3, and the left-hand clade in Figure 6.16. Another adaptive radiation occurred after the Cretaceous-Tertiary extinctions at 65 million years ago, the Cenozoic clade shown in Table 6.3, and the right-hand clade in Figure 6.16. This more recent clade radiated into almost all the adaptive zones utilized by the older clade. However, the Cenozoic clade has become much more diverse in terms of species number. Table 6.3, having radiated into the dung habitat more extensively, e.g., Aphidianae, Scarabaeanae, with the advent of the radiation of mammalian herbivores 55 million years ago. Another scarab radiation was coincident with the extensive radiation of the angiosperms about 50 million years ago, enabling entry into a new adaptive zone provided by the roots, stems, and leaves of flowering plants. The extremely species-rich Melilondinae, with an estimated 10,000 species worldwide, includes the May beetles, also known as June bugs, chafers and their larvae, the white grubs, many of which have become serious pests of pastures, lawns and agricultural crops. The microbial faunas in larval fermentation chambers of scarab beetles have been less studied than those in the roaches and termites, so we do not know if there is a specialized gut fauna, or how it is acquired. Possibly, colonization results simply from the microbes in the substrate, because these communities are likely to be speciose, with potential symbionts occurring in high numbers in rich substrates such as dung and carrion. The persistence of the mutualistic relationship between scarabeoid beetles and a fermenting microbial fauna forms a common thread through two radiations with similar results, the Mesozoic and Cenozoic clades. The breeding systems in the species have changed radically from subsocial groups like the Pacillidae, Mesozoic clade, to a solitary lifestyle in many dung beetles, to parental cooperation in some Scarabaini, e.g., dung rollers contained in the Cenozoic clade. The subsocial breeding system may well have contributed to the origin of the group and its diversification, but the mutualistic ecology of the group with microbes has been instrumental in this spectacular adaptive radiation. It remains almost a paradox that on one of the least nutritious substrates, wood, the largest insects grow, the record being a 205 grams grub of the rhinoceros beetle, Megasoma aictian, Acorn 2007. Undoubtedly the credit goes to mutualistic microorganisms in the external rumen, and as Acorn 2007, page 63, says. Table 6.3 Diversification in quote. Isn't it interesting that all the real contenders for the largest insect title feed on rotting wood? 6.7.4 Ants and mutualism. The fourth adaptive radiation involves the ants, which is no less impressive than those in the Dictyoptera and Scarabeoidea. In fact, the ants constitute one of the most dominant elements in terrestrial faunas, Hodiericus LL. and Wilson 1990, Davidson et al. 2003. Ants are a ubiquitous and dominant feature of the terrestrial landscapes, playing key roles in symbiotic interactions, soil aeration, and nutrient cycling, Moreau et al. 2006, p. 101. Present estimates place the emergence of ants in the late Jurassic about 150 million years ago, but majoritiation occurred in the late Cretaceous to Earliocene, from about 100 million years ago. This radiation was coincident with the origin of angiosperm-dominated forests, Moreau et al. 2006. Accompanying the angiosperm radiation was the expansion of herbivorous insects into tree canopies, such as hemipterans and coleopterans, providing both food and mutualists for the ants. Indeed, Davidson et al. 2003, page 969 found strong evidence that arboreal ants feed principally as herbivores of plant and insect exudates. Extrafloral nectaries on plants, and food bodies from myrmecophytes, plus hemipteran trophobionts, as well as microsymbionts of ants, probably all play important roles in ant nutrition and the radiation of the group. The microsymbionts of ants are housed in mycetocytes, specialized cells with symbionts therein, along the midgut lumen. They are maternally transmitted proteobacteria, and probably contribute to the ant colony's nitrogen budget, Sauer et al. 2000. The assemblage of symbiotic relationships of ants may well have contributed to their abundance in lowland tropical rainforest canopies where they constitute 94% of all arthropods when fogging samples are taken, and 86% of the biomass, Davidson et al. 2003. Mutualistic relationships of microbes with ants include myrmecophytes, trophobionts and Table 6.3 Kant. Gut microsymbionts. In addition to these multi-species complexes, the leaf-cutting ants are involved with their fungus garden mutualists and parasite-inhibiting bacteria. These bacteria produce antibiotics which inhibit fungi parasitic on the fungi ants cultivate for food, Curry et al. 2006. The bacteria are accommodated in cuticular crypts with exocrine providing nutrients, a mutualistic relationship that must have enabled the radiation of leaf-cutting ants a long time in the past. This occurred about 4 million years ago, according to Hodiericus LL. and Wilson, 1990. The persistence of antibiosis after millions of years of coevolution of ants, fungal cultivars, parasitic fungi and the bacteria is impressive. Yet, how does the ant bacterium mutualism avoid the evolution of resistance in the parasitic fungi? The radiation of ants constitutes one of the most impressive adaptive radiations on Earth, Hodiericus LL. and Wilson 1990. It cannot be denied that mutualistic associates have been important in this radiation, but whether they have been critical and fundamental can be debated. It is interesting to note that, as with the Dictyoptera and Scarabeoidea, the ants were originally, and largely remain, soil-dwelling, where they forage in the soil and rotting vegetation on the ground, Hodiericus LL. and Wilson 1990. In a brief consideration of the reasons for the success of ants, Hodiericus LL. and Wilson do not include mutualistic relationships, but the arguments developed since their publication may well provide a compelling scenario for their importance, e.g., Davidson et al. 2003, Curry et al. 2006, Moreau et al. 2006. G. LARECDAE. Many other adaptive radiations have resulted from mutualistic relationships. Consider the diverse taxa of pollinating bees, moths and butterflies, and other taxa such as beetles and flies, tables 6.1 and 6.2. The accompanying mutualists, be they angiosperm plants or microbial associates, have also radiated, with microbes and insects often showing cospeciation and probably coevolution, e.g., Norris. 1979, Curry et al. 2006, Sauer et al. 2000, Aksoy 2003, Clark et al. 2000, Figure 8.9. Overall, mutualism has played a very creative role in the generation of biodiversity and the expansion of ecology into many fascinating areas. Many of the criteria listed by Voucher 1985A, page 23, needed to demonstrate that mutualism is the major organizing principle in nature, have been met, establishing at least that mutualism has emerged as one of the few most important creative interactions in nature. 6.8 Modeling mutualism. Models of mutualism have a relatively long history in ecology and have emphasized several different aspects of relationships. Population stability, benefit and cost, and mutualism competition models are a few examples. As models usually do, they attempt to clarify a conceptual